saying that when Watson and Crick pulled together the double helix structure of the DNA molecule, part of their major stumbling block was how does this stay uniform in width from top to bottom of the DNA? If you took a DNA molecule out of your body, it would stretch, and you stretched it out, it would stretch probably the length of your arm. Well, it's double-sided, and it has to remain uniform from one end to the other. So the way it does that, and this is very important, is you always have a nucleotide that has two rings bound on the other side to a nucleotide with just one ring. So this ring here having one would be bound over here having two. So from top to bottom, there is always one purine bound to one pyrimidine, and that makes for three rings in the center, and that means that the double helix remains the same width all the way down. This is really important because you can't have it wide at the top and go in like a waistline and then out at the bottom or be wavy. So it has to be uniform. And that means that they have to be bound in such a way to have three rings in the center all the way down. Erwin Chargaff was the man, and I think you'll know him later, but Erwin Chargaff was the man that was looking at the molecule and determined that this is the way the bases align themselves. So he determined everywhere he saw an adenine, he saw a thymine. Everywhere there was a cytosine, it was bound to a guanine. So he devised some base pairing rules for DNA, which said A is equal to T and C is equal to G. And that meant that if A is equal to T and C is equal to G, there's going to be three rings here in the center, and the two sides are going to be equidistant apart. Now, if you notice, you see this 5' to 3', and we'll talk about it on the next slide and why it has to be there. All right, your nucleotides are linked together to build this polynucleotide, and then the polynucleotide is either going to be RNA or DNA. But your adjacent nucleotides are joined. Now, when you have the phosphate group sugar, they're combined by a covalent bond. It's called a phosphodiester linkage. And the linkage consists of the phosphate group that links the sugars of the two nucleotides. So these links create the backbone, and then the bases are going to be in the center. The sequence of bases for DNA and RNA is unique for every gene, so it's not always going to be the same. That just makes sense. You wouldn't have the same recipe to make a pie as you would a cake. And so depending on what protein it is you need to make, whether it's a hormone, whether it's an enzyme, whether it's a neurotransmitter, whatever hormone is going to have its own sequence. And that means that the bases are going to be different. All right, your DNA molecules have got this spiral. We know it's called the double helix. And the backbones, the two sides, they run in the 5' to 3' direction from each other. And so that means that they're anti-parallel. One DNA molecule includes many genes, and when we get more into the DNA molecule a little bit later, we'll find out that when it transcribes itself or when it makes a copy, it makes a difference in whether it's being copied from 5' to 3' or 3' to 5'.